maybe related to the way the diagnosis happens, and given that it's a disease of old age and the concept of that you get lots of problems with the aging process, probably there is much less awareness about the disease, and we probably are losing many lives to this disease, being just unaware of that, of the fact that there is a treatable tumor in there which can be easily treated if can be diagnosed. So I personally believe that having a better um, awareness of the disease can really change the face of men's health in India as well. And we have to be very careful about propagating and making our population aware of different types of cancer. India has done pretty well with respect to advertising breast cancer, but somehow, it's men's thing probably not to talk about the diseases, and this is common all over the world. But I think the breast much better than what India has embraced. So the research, of course, is driven by where the demand is of the research problems, right? So if prostate cancer has not been identified as a common research problem, the research funding and the research directionality is not going to be there as well. So in my belief, the first concept is to put the awareness out there and get the real data on how much this disease is causing harm to economy, harm to our population, to our patients, and how good it is to rescue them from these difficulties. And this is where the research direction definitely need to move towards.
Right, right. Yeah, well said, Jyotsna. Jyotsna, if we see, 5 years ago, organoid models have really emerged, especially in
Mhm.
cancer research. So,
Yeah.
and I believe, that you have also developed, some of these, towards prostate cancer research. Would you please tell something about the organoid models, especially have been preferentially used in prostate research?
Yeah, absolutely. I mean, the first prostate organoid model was actually published by Hans Clever's lab in the USA. As usual, all big stuff comes from there. Actually, this one was from Sloan Memorial. Um, and so in our case, we were really lucky enough that the clinician scientist who was involved with this actually belonged to our institute itself. Um, so he did his PhD with my mentor, Professor Judith Clements, and then moved to Hans Clever's lab, and then got back to to do his clinical practice. And this is what actually led us to really venture into the expertise he has honed while he was in Hans Clever's lab. And being a clinician, he is really keen into bringing the research into clinics. So we have been continuously involved in now since then after his return in developing organoids models, and that's only possible because we are just based in a hospital campus itself. So hospital is just where the urology department is, just 5 minutes walk from us. We have access to all fresh tissues, which has been instrumental to develop organoids. So we have been able to develop some very specific organoids within our group, but we also have access to the ones which have been developed by Jan Vella in Sloan Memorial, and he brought them brought those with him when he moved to Brisbane. So that also gives us a very quick and easy access to these organoid cultures. They're pretty expensive to grow because they are dependent on a very rich and specific media, but the outcome from these could be so much closer to what you observe from patient samples. So we still tend to do a lot of analysis using these organoid culture models. And one of the recent work which we performed was to looking at the PSA genetic variants, where
Right.
we expressed PSA in its wild type form and its variant form in these organoids. And the outcome you see is quite different from what you see in cell lines. While cell lines are just so homogeneous and you see them coming straight forward, it's a little bit complicated when you start working with the organoids. But it gives a little bit more reliability to your data, that what you're coming up and getting the outcomes more translate more close to the patient outcome. We also do use these organoids quite a lot for sequencing. So we grow them in mice, and recently, one of the patient was actually not identified to be BRCA positive in his initial sequencing analysis, but after growing organoids, we knew that patient was actually going to have it had probably rare BRCA mutations, was missed by the tissues sequencing itself, or somehow not picked up. But by doing his organoid culture, we were able to check that, and he was eventually put on to PARP inhibitors, and his life could be saved. Otherwise, his tumor was growing really aggressive, and we had no clue which direction he's going to take. But so these sorts of culture have really helped us even to change the patient care, not just doing the lab-based experiments, which has been very terrific outcome.
Right, right. But there's also a very sorry figure if you really look into the Indian conditions, Jyotsna. Not many are really insured, in terms of getting diagnosed or even getting operated and all these kind of things. And with a kind of an inherent stigma prevailing in Indian conditions, where do you see the ray of hope in development of some cost-effective diagnostic measures, for prostate cancer in so-called developed countries, and yet making up some kind of cost-effective measures, like for example, maybe in conditions like India, many men are aged above 55, they might have some kind of inherent stigma, and yet they might have some extensive factor in approaching the doctors. If they have such kind of scrotal pain or all these kind of things. Do you see something like a kind of a pregnancy kit kind of thing that is really coming up in some countries like India and Africa, where
Prash, unfortunately, you've been breaking up a lot. So, um, are you going to repeat your question, please, so I can actually understand? Hello, hello.
Ma'am, please wait. I think again they are not audible. Their connectivity is not clear.
Hello, hello.
Ma'am, please wait.
Okay, yeah.
Yeah, Jyotsna. Yeah, I'm sorry. I could hear everything excepting the last sentence. Please go ahead. Yeah.
Prash, I think you need to repeat your question because I could not really hear your question. The last one, as far as I understand, it was more related to um, men's health, is it?
Yeah. I mean, what I mean, to put it in a very short way, Jyotsna, I know there is a lot of inherent stigma, prevailing in Indian conditions. So, do you
Yeah,
see a kind of a pregnancy kit like thing, coming up, for diagnostics, probably at home, maybe a simplest
yeah,
kit kind
yeah.
of thing, for prostate in men? Yeah.
Yes. Yeah, absolutely. I mean, I would say that this is not just in Australia, in India, but the same problem we face everywhere. Men are so much resistant to talk about their health and not go to the clinics as often. Probably women are ready to go and embrace the conditions and talk about it as well. So this has been I actually was involved in doing a survey of Australian men just last year under a program to actually put our own PSA test as a home-based test kit, and it was absolutely amazing to see that um, the women, actually, the partners who either had prostate cancer or not even, were so keen on having these tests to have their at home ready. So in Australia, we actually do a home-based test now. We do a bowel cancer screening. So anybody above the age of 60 is sent a kit to do testing for bowel cancer. It's a home-based test. Even if the result does not come at home, but you can just take a sample and post it. And if there is any thing, you could be contacted and asked to come to the clinic. So at least something like this would be really useful, but we have to always think about the cost effectiveness and um, uh, which is what is possible and what is not possible in a current economic environment. So in my opinion, awareness is the first and foremost thing. And if we could develop something which is a home-based kit, would certainly would also be helpful for men to take this test forward and come to the clinic. And actually, the early diagnosis is the key to any kind of cancer, and that's true for prostate cancer as well. I have been hearing so many cases around coming from India where, my friends, relatives have been affected by
Yeah,
the disease, or someone other than that. I mean, these were not
yeah,
used to be
wonderful,
common.
Jyotsna.
Yeah, absolutely. So, this would be a terrific outcome if we would have a kit, which is not that difficult to develop. I think all of we have lots of good research going around and can easily develop, especially for PSA testing. And then back to its implementation and how to make the population aware of the disease and make them go through this test, even if it's a home-based test.
Right. An excellent, wonderful insight, Jyotsna. So, Jyotsna, you also have extensively worked on non-coding RNAs, primarily on microRNAs. The last 5, 6 years, you have worked on long non-coding RNAs. Some of them are intergenic as well. Do you see some of them as plausible players for biomarker development in prostate? Yeah.
Oh, absolutely. Yeah, I am a firm believer of any sort of um, non-invasive testing, at least as a first pass. And microRNA, given their stability in plasma, seem to be one of the key candidates, right? So some of our own work has led to some of the patent filing. The difficulty which I can think of, and I have been trying to really commercialize these, because first thing what we face difficulty again is that the bigger the sample size, the robust outcome it is. So I am really keen on replicating it, the panel what we have got, which actually also involve some of the microRNAs previously identified by other groups. So some of these, like MIR141, I think, is has come up in every possible analysis coming out of different labs around the world. So it does make sense to take these forward. But the reality is probably a bit different from the concept of taking them forward just theoretically, right? So, what I have seen is that pharma companies don't seem to be much interested in diagnostic test until it also leads to it's something which is associated with change of the therapeutic regime, and is also a little bit driven by commercialization process, which does not look like a very conducive environment for the pharma companies at this stage, which are much more interested in pharmacogenomics rather than just diagnostic or prognostic biomarkers. And a lot of difficult um, thresholds right now. I mean, if I look at these thresholds, I don't think PSA test could have ever been approved if we go by the current criterias for taking any new test into the market. So some of the tests are already in there, even microRNA based coming up, urinary long non-coding RNA based, for example, PCA3 test is already up there. So we are making progress, but the key to this also is that we need to come up together, make them into a very robust analysis, do the analysis, putting the data together, and make it so robust that you can have a clear cut answer of using them at the commercial level. And I think that's the success to any key to success to any research program.
Yeah, perfect. Wonderful, wonderful, Jyotsna. So moving further, Jyotsna, how has COVID affected your research? Could you please tell us? I mean, has it really affected your research? Being in Australia, your research group, your research allegations. Yeah.
It certainly has. I mean, we were almost in a lockdown phase of 3 months in the first wave, but Australia has been very lucky that it has been able to so far control, and Queensland, where I'm based, is still enjoying the kind of COVID-free environment at this stage. After once we open in July, but March to July was the phase which was when we were in almost complete shutdown, no lab work available. We try to continue with research as much as we could by offline stuff. A lot of my lab-based research is part more lab-based. A part of it is also bioinformatics based. So it did not affect those as much who were doing in bioinformatics research. But the ones who are completely wet lab-based were feeling really affected by it. It has certainly slowed down the research, and so is the funding as well. And it has been quite detrimental for the universities because we have lost lots of revenue coming out of the students and international students, which then directly affect us as a researcher as well. Lots of jobs are happening too. We tried to stay as much positive as we could and tried to also use our expertise for getting some outcome for COVID-19 research. And I'm so glad that you could also be part and do some bioinformatics and genetics based analysis of identifying key pathways involved in a specific population affected by COVID-19 with severe first phase outcome. So there has been lots of negative effects, but again, as I said, that it's all about making best out of what you can in that situation, and this is what our lab has tried to. And we have been also very lucky that we have been able to enjoy a COVID-free environment for a while now, and keeping our fingers crossed that we don't have to face it again. Another big impact has been certainly on travel. So a lot of the collaboration which could have generated could not be generated at this stage because of no travel. But again, we can see the positive effects like having this Sci'versation with you, and probably would not have been happening if there was no lockdown. So there's always positive about things. Yeah.
No, perfect, Jyotsna. Yeah, that's been amazing. One thing I always wanted to ask you, Jyotsna, that as a women researcher, you might have faced a lot of challenges. Right from journeying, from your PhD heydays, to your postdoc and your early career research program, and now as an associate professor and a wonderful group leader, and as a prostate cancer researcher and one of the topmost, cancer epigeneticists of the world. If you really go through your research over the last probably 18 years, what is that thing that really touched your heart? Maybe what is that kind of hurrah moment for you over the last 2 decades of your research career? Yeah.
I'm not sure if I would define this, but I get so excited about anything new we find in our lab. So for me, like at least every week, I get a hurrah moment, and that keeps me going always. Yeah, I mean, most of the time, my students will come and say, "Oh, we found this thing doesn't look right," and I would say, "To me, this looks most exciting when something does not look right." But yeah, let's just do it again, and if we see this again, um, so this is actually, I think, the best part. There are, of course, certain additional things. Once you get your first grant, your first fellowship, they really add lots and lots of value and confidence in your own research. Getting promotions, and which is not easy, again here. And as you said, also being women, I have to, of course, worked hard and probably double, and also not in your own country. That also adds a little bit pressure. I mean, I haven't seen a lot of um, um, discrimination within our department, but off and on it does come a little bit here and there. So you have to take care all of that into account, and again carry on with and suppress any negativity with your hard work, and which has been happening very well in my case. And the hard work always speaks up, and this is what has been happening. So a lot of my hard work during my PhD, which led to a huge number of publications and led to a good outcome with respect to my postdoc career. um, I think good one thing which actually I probably want to mention, which changed a lot of the definition and the way how I could get to the position where I am, also include persistence and hard work. But the changing kind of moment in my career was that when I actually joined um, as a postdoc fellow to start my career in prostate cancer. Within 15 days of my joining, my guide and mentor actually left the institute. Her husband got a job in Switzerland, so she moved with him. So I was so much lost when I came and joined here. I mean, I was changing from asthma to prostate cancer, a new country, a new environment altogether. Um, yeah, I mean, I was just completely lost initially, but that was the moment when I actually in my research career. So that was kind of a very big moment as well. That not having a real a mentor. I always say that I was adopted by my that time mentor, who actually had no genetics background. So in fact, in my whole institute, nobody was doing genetics at that time when I joined QUT. So I have to find my synergies, my collaborations, all by myself from scratch. I did end up meeting some mentors down the track who were very helpful to shape my career. But I think this was one of the key event as well, which I consider that which made me quite independent from the very beginning, and also made my thinking quite independent, because I had to start making decisions. I used to be so confused that as a postdoc, how much authority do I have to make decisions with respect to everything? But yeah, it did come eventually and slowly with time. And I mean, I'm one of the first ones within the QUT, for example, to receive Cancer Australia funding. I don't remember I was able to get any successful NHMRC scholarship grant application as an example, because they were used to be so rare within my university when I joined it. So a lot of things have come through with hard work and persistence. Um, of course, you face difficulties, but again, it's all about how you overcome those and carry on your career, which has been wonderful in my case so far.
Yeah, absolutely, absolutely. I'm absolutely in awe, Jyotsna. Yeah, thank you so much. But one final question to you, Jyotsna, before we move on. As a successful scientist, you might have disagreed in an agreeable way, with your colleagues. That's way, you might have, moved forward in bringing out, successful projects. Can you come up with one such instance where you probably agree to disagree on certain stuff? Maybe it could be anything, in deliverables or in making up your publications or whatever. This question is being posed to all my guests. That's because the current generation of young scientists probably lack this kind of approach. Maybe I really wanted the young brethren to take inspiration, an extra inspiration from such words from wonderful scientists like you. Yeah, please. Yeah.
Um, if I understood your question correct, is that the how to handle the controversial situation? Is that the question? And
Yeah, it need not be controversial, Jyotsna. It could be anything. Certainly you might have disagreed in an agreeable way. It could be anything. Yeah. It need not be a kind of
yeah.
a controversial thing, but maybe with your senior colleagues or with your ex-PI or with your co-PIs, maybe if this experiment could have been done in this way, or we might have met this particular deadline a priori. There could be some kind of such
Oh,
instances.
oh, yeah, there has been lots, they always have come up, even during the PhD and the later on career. I mean, especially my PhD was very interesting. With Dr. Balram Ghosh, initially we had lots of conflicts, but in my case, I just realized what they wanted, and how can I do the thing what they wanted, but my own way. So I think you need to really recognize what is what is required, and there are always ways to um, turn things in your favor most of the time, and also get your point across. One of the example I want to give is actually was with my own PhD student who I thought was absolutely not capable of doing PhD because she wasn't ready and has not got the skills. Um, but eventually, we did come up into terms that she can culminate by finishing it as a master's degree rather than completing a PhD. But it took lots of energy from my side to also from myself. But I think was in her favor that she could do much better job with her master's degree and after PhD if she was not going to have a career in academia anyways. And it's a difficult market situation as well in Australia to get jobs for PhDs in comparison to what you can get for masters. So there has to be some sort of balances. Like I did not also want to spoil her career from the perspective that she just end up by not doing anything after coming. But then at the same time, if you're not intellectually sound and you're not being able to produce the results, it's the most important service we do is to science, right? That is what we need to always keep in our head, that science has to happen very ethically and very logically. Um, so to do that, sometimes you have to make tough decisions in your life. And I have been almost up with it. I had never had any problems with respect to um, publications and stuff. I always have got colleagues which are very collegial and collaborative, and I think this is how science works. And um, if the goal is to get good research outcome, we can work more collegially than of having difficulties in going in the same direction. And that has been the case with the practical consortium, where I think now we are more than 100 authors on these papers, but we have been through lots of discussion how these will be handled, and it has been working pretty well. Everybody get their speciality contributed, and then the data sharing process happens. So it's all possible to do everything as far as you're talking.
Yeah, yeah, awesome. Yeah, well said, Jyotsna. I think this is certainly kind of a need of that, and I know it's not that easy to become a wonderful scientist in making, especially after having gone through several phases in your career. These are all parts and parcels of life, especially the research life. Disagreeing in an agreeable way and other stuff. So, where do you see yourself the next 10 years, Jyotsna?
I truly consider myself as an academics, yeah. So truly speaking, if the COVID situation would not have come through, I would have already been looking from my career perspective to have something further next as a next step of getting another promotion as my personal career. Um, but where I see myself next in next 10 years is actually nurturing the young generation. So I'm having to have my skills passed on to next generation and want to see them flourishing and making their own labs all over the world, and also being able to commercialize some of the research and really doing the research which is much more translational rather than keep on doing little bit things here and there for the sake of publications. I don't think that's what my long-term goals are. You have to do some of those things initially, but when you are establishing yourself, because you have to be well known, there are some loopholes within academia. The success is measured by a lot of different things, such as publications and your success with the funding. But the ultimate outcome is at least what I see myself in 10 years is that if I could really do something and get something out for patients, better outcome, whether it's prostate cancer patients or any other cancer patients, and that probably would be considered the best success for myself alongside taking the next generation to the point that they get ready to contribute to the betterment of science and can do science really ethically and in a manner which is inspiring and which is translational.
Yeah, yeah. Well said, Jyotsna. So, if you were to be offered a kind of a position in India, if at all, if you were to consider, to come back to India, so which research institute probably you would opt? And I'm sure, you would definitely, think of, leading a big lab and becoming the director of an institute, which India should
Yeah.
always be proud of, having, someone like you, at least, people like me would, would like, you to be in a kind of a premier institute, as a director. So, do you have any takes on these things, Jyotsna?
Um, in some ways, yes. In some ways, no. Because I'm really passionate about researching on cancer and doing genetics research still, yeah, and that's where my expertise lies as well. So, if I ever get an opportunity where I have the possibility of leading these sorts of areas, this would be my priorities. Um, I actually maybe also have a little story to tell here that when I was finishing my postdoc position, which was only for 1.5 years, I actually tried to get back to India, but then the whole planning changed and it didn't even work out. But currently, I have been thinking on these mostly because so that the directionality of the research can be changed and can be improved if I can go back. But I'm also happy if I could work as an adjunct in collaboration with great scientists like you and continue some great science for the time being. But I just do not want to name an institute. I have been offered from few really good institute the positions, but at this stage, I'm still considering whether I'll be returning back or will be staying in Australia for a bit more time. If I want to leave Australia, I want to leave it with a mark as well. Like I don't want to leave so quickly that people forget me so quickly as well. So I aim to stay here for another 5, 10 years first to make it a substantial contribution. I think I've been doing pretty well, all right, but something which can be remembered and can be taken forward. I always keep that photo of the science in front of me. So once that happened, I think I will be very happy to consider my move and keeping my research interest intact as well.
Yeah, wonderful. I mean, as on a pretty lighter note, as someone who had been to Commonwealth country, returning back to probably Commonwealth country like India again, that too, your alma mater, something your own country, that would be a wonderful accomplishment, Jyotsna. India would be absolutely be honored to have a wonderful women scientist like you. We are absolutely inspired, motivated, by your wonderful research, passion, and most importantly, your smile, your wonderful, research navigations, the way you really look at science, and most importantly, the way your project is really, taking up. Someone like you, sitting in Australia, leading a kind of a translational research institute in Queensland University of Technology. That's been absolutely awesome and incredible. We wish you all the best, Jyotsna. May you keep inspiring, as many people as you could. Do you have any final question for Bioclues members or any young brethren where you want to give or offer any advice?
Thank you so much, Prash, for all these nice words. I think whoever I come in contact with, they become inspiration for me. I've tried to take good thing from everyone whom I see, and probably this is what I want to also give as take-home message. You cannot, of course, um, beat hard work. If you are working hard, you could achieve something. But I always want to use that, do the hard work with some smartness. If you just keep on doing hard work without using your brain and just physical hard work, that does not lead to anything. Keeping abreast with all new research is most important. I see this with the young generation. They do not like to read papers. They would just want to glance over things, just go by the quick information which is possible, which sometimes lead to lots of misunderstanding. So my rule have been from the very beginning that I do not sleep before reading one paper at night. Yeah. And you see the accumulation, it never ends, right? If you keep on reading, it just adds value. So I'm a big fan of that you should be reading papers. I mean, at this stage, probably it's not really required to read full paper for me, because by reading extracts, I can now find out what is happening in this paper, where are the problems. But I'm just talking about my PhD career and my postdoc career. I haven't let a single day go when I was not reading a paper, at least one paper per day, and full paper, not just the few things on it. That is probably one of my other advice, that um, knowing what's happening in the world in your research field is really important, and changing, if possible, some of the time the direction to find something new and novel is also the key. A lot of the time we are tied up by where the funding takes us, but if it's possible sometimes to do a novel research, not just repeating others' work, is the key to finding new things. Yeah. So innovation is the key. That's my probably last advice. Keep the young mind innovative all the time, and carry on research with passion and rigor.
Oh, wonderful. Yeah, so innovation is the key. That's a key take-home message from Jyotsna Batra, who is our guest today. A wonderful woman scientist, always smiling and always inspiring many young brethren. And one important thing, especially the PhD fellows who are really watching over here, you have seen Jyotsna Batra of 2002, who was a fresh MSc graduate who stood top, as a topper in CSCR net examination, and then she went on to gain, wonderful, percentiles even for GRE as well, which helped her, move forward, as a successful researcher in Australia, and exactly a tacit PhD, a wonderful PhD with Dr. Ghosh in IGIP for about 4 years, where she produced 17 publications. Yeah, which is quite fascinating, amazing. I think, friends, I think this has been the best in Sci'versation so far. Although we had some intermittent net connection problems, I'm sorry for that. But Jyotsna Batra is always there, for any possible advice, I believe. She's she's quite active in LinkedIn, and she's also in Facebook. And you can probably email her for any possible advice. I'm sure she will take a few minutes of her precious time in motivating young generation. Thank you so much, Jyotsna, for your time. It's 8:45 your time, and thank you so much. It's been absolutely inspirational. I'm more than honored, to have you as a guest today. Thank you so much for your time.
Thanks to you, Prash. It was really wonderful talking to you. And I look forward to strengthening our collaboration further and going on with our cancer and research.
Thank you. Yeah, thank you. Thank you so much, Jyotsna. Yeah, bye. Yeah.